try with this.
We're gonna restart.
Hold on.
Restart. You should pause it.
Is that Tushar? Is that one?
This is the one that Tushar was using. It's mine.
Oh, maybe you say play. Let's see what happens.
This one?
Yeah.
Oh yeah, it's good. Okay.
You need to look at it.
Oh, oops. Okay. Hey Hannah, do you start?
Yeah.
It's already okay. Okay, so for this part of the talk, I'll discuss the issues that you might potentially have during in-sequence. So some of the common things that you might face is like probe efficiencies that vary, and the other issue that you might face is background. And so based on our experience doing Flish experiments, we have tried to understand the potential reasons why you might get background and the steps that you can take to like eliminate or reduce background in your experiments. And we've also come up with like a few approaches that you can use to validate in situ in your mouse or human tissue samples. So background. So we broadly classify the kind of background that we see in our experiments as templated or untemplated. So untemplated background usually arises when you use like high concentrations of probes, and they tend to like either stick to one another or clump to one another, and also adhere to the surface of your slide. So so we've seen that they can attach to like the coating on the slide. So slides that we use usually to collect sections have a positive charge, and the probes are like oligos are usually have a lot of negative charge. So they have a tendency to stick to surfaces of cover slips and glass slides. So that's one kind of reason why you might have like high concentration of probes sticking onto surfaces, clumping, and they might like initiate circle formation RCA, and you might end up seeing something that looks like puncture. So this is going to look like puncture, but you mostly see it distributed in acellular regions, like regions which have like no nuclei, no DAPI signal, but still show like bright puncture. And when you take an image of your section, of your sample in a confocal microscope, and when you scan through the different Z-planes, you will see that the puncture that you get from untemplated background is usually closer to the glass surface, which is usually where the probes clump and result in rolling circle amplification. And this phenomenon is more common when you use like more than 10 sets of probes per gene. So that's when you're going to use like each gene at like certain amount of concentration, and if the concentration is going to exceed 1,000 nanomolar, this phenomenon occurs more often. So this becomes a problem if the transcript that you're looking at is already rare. Then you're not able to distinguish whether it's like low expression or is this no expression. And this also becomes a problem when you look at like uniform cell populations, because you would see, say for instance, if you're using cell lines, if you see signal in every cell, then you don't know whether this is real signal or is it like real signal plus background, which you're just interpreting as signal. So some of the things that you can do to like either eliminate or reduce background that arises due to these features is always choose your H probe or the hybridization probe. So let me go back. Sorry about this, but I think I just want to run through this. Okay, so this probe, the H probe or the hybridization probe, has the region that's going to actually bind to your target mRNA. And this is the part that you need to like choose more carefully. When you design your H probes, you need to choose them in such a way they have like optimal TM. So we recommend a melting temperature of around like 45 to 65. So we'll go over like in detail about how to design H probes in the probe design session. That's going to happen after lunch. But as long as you like stick to the optimal design protocol and pick a suitable PM, so a suitable TM, you should be able to avoid sticking of probes to random non-target sites.
Monica, I have a question.
Yes.
In terms of the non-specific
explore like a high background
Yeah.
genes. Like for example, when you do immunofluorescence staining using antibodies, like different cells
Yeah, yeah.
actually has different background. Do you have the same issue using the probes? Like different cell types has like some cell types have higher background, maybe noise. others.
Oh, so no, so we have noticed that untemplated background is usually uniform because it really doesn't like depend on the kind of cell type you're looking at. It usually binds to the cell glass surfaces because of the charge. But based on literature pair also like things that it might bind to like lipids. So when you take a section, you have like cellular material that gets deposited on the slide, and probes might have like a tendency to stick to some more often than the other. So depending on like what tissue that you work on, things might be a little different. But usually if you use a probe which has like a reasonable TM, non-specific binding would usually be eliminated. And the other thing is, so when you use abundant transcripts, like genes that you know are expressed in like really high, have like very high FPKMs, you would stick to designing like 5 probe sets. So you design like 5 sets of probes that tile on the mRNA. So that's usually good. If you're working with something that's like moderate or rare, then you want to like use a few more so that you improve your efficiency of detection. In that case, you design up to 10. So the way we use these probes is we use each probe at 100 nanomolar concentration. So if you were to use like 5 probe sets, there's a left and a right for a set. So that's going to total to like 1,000 nanomolar here. But when you're going to design like 10 probe sets and you're going to use them at 100 nanomolar, the pool concentration is going to be very high. So in this case, what you would do is you work it out in such a way that the pool concentration does not exceed 1,000. So if you were to design like 10 probe sets, then I would use them at like 50 nanomolar, so that the total concentration stays under 1,000. So this is, and even if you, despite using them at like 100 nanomolar, this concentration is already like pretty saturating, so that should be like good enough for the thing. If you go even below this limit, then you might have to incubate your edge probes for a longer duration. So we usually do it for like 2 to 3 hours. But if you go like even concentrations that are below 50 or 100, then you might have to incubate longer. And the other thing is, so stringent buffer washes after your edge probe hybridization and ligation should also eliminate untemplated background related issues. So initially some of our buffer washes used to be in PBST, but now we've replaced them with a different kind of buffer, which is more stringent and helps to eliminate non-specific binding. The other, another approach that you can use to eliminate untemplated background is you use truncated edge probes. So these are edge probes which would have the mRNA binding region, but lack the region that's going to initiate the circle and the bridge. So when you use these, or you use scrambled oligos, they would just adhere to all kinds of sticky surfaces, which tend to like attract oligos. So you can use this like a pre-hybe, saturate all those non-specific sites, wash it off, and then add your actual hybridization probe.
Yeah, I have a question. So you mentioned that, the probes will stick to the size.
Mhm.
Do you guys block size first?
No, so that used to be a problem in our initial experiments, but when we started using the current stringent buffer, this happens like very fast, so you should be good. Unless like you use like if you're using like pre-coated slides commercially available, I think the coating is more or less uniform. But if you're gonna do it yourself, which you might do in some cases like cover slips, so in that case it's hard to kind of control how uniform it is. You might see some like blobs of signal, but there you can usually distinguish it because they're going to be like in acellular regions very close to the slide. But with this buffer, it eliminates like most of not probe sticking to like weird surfaces.
Yeah, and then another question is, I think this method will detect the signal at a single molecular level,
Yeah.
right? So I'm just curious why it will, relates to the gene expression level, and the moderate or high expression level, since this method will detect the single molecular level.
Yeah.
So in theory, they should have the same chance for each molecular. It doesn't matter what's the expression label, right,
Yeah.
for this gene.
So not all probes work the same way. So I'll show an example where like you design, say for instance, you had a gene of interest and you designed like 3 or 4 sets of probes,
Mhm.
and you test them individually, you will see a lot of variability in how one probe works versus the other. So that's got to do with like what is the accessibility of the probe to reach that mRNA binding region. It could also be an inherent problem with the probe because it tends to like form hairpins or self-annealing sites that it never gets there. So that's one thing which we have noticed. The other thing between moderate and like abundantly expressed transcripts is, in abundantly expressed transcripts, the probability of the probe that you add binding to the mRNA is very high. So it's you detect it more often than not. But for rare and moderately expressed transcripts, we usually use 10 probe sets so that the chance of detecting them is higher. The other thing is we, at this point, don't have a measure to say what is our efficiency. Say if you had like 100 transcripts and you detected like 70, that's very different from having 10 transcripts, like the efficiency of detection.
So can I say, for those genes that express very high label, and then you use 5 processes,
Yeah, we
then
usually
maybe
use
you will
5.
miss some target genes,
Miss some.
targets.
It's possible. We
Right.
haven't, so there's no we don't have like an estimate that a cell has so many transcripts. We kind of base it on FPKM.
Okay.
So this is being done in great detail using cells, which Per was showing you earlier, but we haven't like correlated to how it looks in tissue. Yeah. So with moderate and rare transcripts, we usually stick to using more number of probes. The other kind of background that you might see is templated, and this usually arises if you have partial complementarity to non-target transcripts. So I previously told you that the hybridization probe has a region that binds to the mRNA, and choosing this when you choose this region, you need to be careful that it does not have a partial complementarity to any other mRNA in the tissue of interest. Okay. And this is going to look like true signal in cells. This would look like puncta in cellular regions. So this again becomes a problem if you're looking at like relatively low levels of transcripts, or when you're staining uniform population like cell mix. So some of the things that you can do is again pick optimal TM. Don't go for regions that have like low melting temperatures. And the most important thing is always blast the sequences that you're going to use for designing probes. And this becomes very important if the transcript that you're working with has several homologs. So in that case, then it could like bind to partially complementary other transcripts. And so these two usually could take care of templated background along with stringent buffer washes, and after the H probe hybridization and after ligation should eliminate some of these. The other problem that you might face is if your H probes, so when you design H probes, you always look for parameters like it should have an optimal TM, it should not have sequences that share too much complementarity to other targets. And you also need to look for potential dimers or hairpins. So when this happens, your H probes are just going to like dimerize or form hairpins, not be able to bind to your target mRNA, and you would not see signal. And in that case, your transcript will still be there, it's just your H probe is not able to go and bind to it. Okay. Yeah, so this is an example where we are looking at an in situ for the LCK mRNAs. So 2 probes were designed at targeting different regions along the mRNA. And you can see that this probe gives good signal. So basically what you see in magenta is the signal. And the second probe does not give good signal. So probes have varying efficiencies. A couple of reasons why this could be happening is, so probe 1 targets a specific region here, which could have been more accessible than this. It's possible that this region has like secondary structures or proteins being bound that prevent accessibility of probe 2 to bind to that region. And the other option is this could also be that these probes tend to form like dimers with themselves or with other probes, and they're not able to basically bind to this region. So now I'll talk about a couple couple of approaches that you can use for validating your probes when you do in situs in mouse or even zebrafish samples. So you can try your in situ in gene knockouts. You can also use like reporters that express your gene of interest and use that for validating your probes. So this you can use as a negative control, and if you know that there is a tree line that's driving the expression of a particular gene in a certain cell, then you can use that as a positive control. And the other thing that you can do is you do in situ for your transcript and combine it with an immunostaining for the corresponding protein. So here I have an example where the in situ where green shows in situ for SPC, which you can see here, and the signals in red is the antibody staining for the protein. And you can see that these cells which have puncta for SPC mRNA also show staining for the SPC antibody. So this would be like one way you can validate. So if you're working with human samples, then obviously you won't have like accessibility to knockouts. So in this case, what you can do is, based on either previous literature or by looking at the protein atlas, you can find out cell lines that have reasonable FPKMs for your gene of interest. You can use these cell lines as a positive control to validate the expression of your gene of interest, and you can use cells with knockdown as a negative control. So this should be like one way of validating it. And if you have a previously well-characterized expression pattern of your gene of interest in specific tissues, like AXIN2 in the liver or intestinal crypts, then you can use those tissues, validate your probes, and then use those probes for whatever tissue you're interested in. And again, you can always do the in situ and combine it with immunostaining. Okay.
So I have a question.
Yes.
If you want to combine with the immunofluorescence staining, which one you should do first? The
Oh,
antibody
so
or the probe?
we do the in situ first.
Okay.
Yeah, because you want to keep the mRNA. So you don't want to do the immunostaining first, because you don't want to like wait till your mRNA degrades.
Mhm.
So you want to first do the in situ.
But you fix the cells or the tissue already, right?
Yeah, yeah.
So,
But I didn't get it.
so you fix the cells,
Yeah,
and
it's
the
good.
mRNA should be fixed. And
Yeah.
you still think that if we do immunization, this and that, that would damage the mRNAs?
It's possible that it could like degrade over time, even though it's fixed. And also, in immunostaining, you might use serum and other components which might not be RNase free. So usually we prefer doing the in situ first, and then we proceed doing the immunostaining. And for immunostaining, we use buffers which have EDTA to minimize RNases, because you use serum for blocking, so
Yeah.
that's not RNase free. Yeah. So this is an example where the in situ was done for BMP-R2 in a cell line, in pulmonary endothelial cell line, which are known to express this gene. So this is a control where there is a scrambled siRNA knockdown, and you can see puncta for BMPR2 as expected in these pulmonary endothelial cells. And when you do the actual knockdown of this gene, you see that there is no signal in here. So this is again like one way that you can validate. Oops. Okay. So any questions so far? So this is the part about quality control and validation. We can go over probe design after lunch, and then we can head to the lab where we can show you how we do the experiment.
Question. So, in addition to this fluorescence probe, is it possible to combine this with the digoxygenin probe in white light?
Oh, okay.
So that to increase the amount of gene that we can analyze. For example, in this case, how many we can analyze with the fluorescence probe?
Yeah.
5 or 6, right? Something like that.
Yeah.
So
You
it's
can do
possible to
5.
add those to combine with the
Oh, so that you can add another extra.
Yes.
Yeah, as long as you can. So the
With
way.
oxygen, yeah.
Yeah, as long as you can conjugate it. So this is the final step where you add the detection oligo, and here is conjugated to a fluorophore. As long as you can have your oligo conjugated to any kind of detection system, I think it should work. Yeah. But in your case, you wouldn't use
I
fluorescence.
think you mean the signal is not fluorescence, right?
It's not
It's not fluorescent, so
NBT,
this, yeah.
APMI.
Yeah, yeah, you just like image it right there, yeah. Yeah, as long as you can conjugate it to oligo, yeah. I don't know if people do that though. Do they conjugate digoxin to oligos? Yeah, but again, you need another antibody, right, to detect it. It's like, more, yeah. Yeah, so using this approach, so far we've been able to image in 5 channels, so that would be Cy5, Cy3, Texas Red, 488, and Pacific Blue that works in 4 or 5. So you can image these, and then add DAPI, and then like basically image it again. So that way you can get like DAPI plus 5 probes. So if you don't want to do 5, then you can always do like Cy3, Cy5, Texas Red, 488, and DAPI. Yeah. And if you were to do DAPI or like add reagents later, you would collect your sections on power slips and use steel chambers so that you can add and remove reagents after you're done imaging. We'll talk about that in detail when we get to the lab. Yeah. And briefly about probe design, so these edge probes, these are the ones that you would design every time you do the experiment. The bridge is going to be common for all fluorophores and all circles. So that's like a one-time thing. You just order the bridge, and that's going to work with all kinds of fluorophores. The circles, we have like about 5 circles that work with 5 different colors: 488, Sapphire, Texas Red, and Pacific Blue. And you would order these. So these are like, we already have the sequences for the bridge, for the 5 circles. So you would just order them once. It's only the edge probe that you have to order each time you do the experiment, depending on what's the target mRNA binding region. Yeah. Any questions before we break for lunch?
Is there any distance we should keep, and should we keep some gap between the different probes?
Yeah, so we usually leave it around 100 base pair. Between them. Yeah, so if this was your target mRNA and I'm designing it like here, I'm gonna like skip 100 base pairs and pick another one.
So then for some, short CBS,
Oh,
some
like,
genes
some genes are
very
really short.
CBS.
Yeah, in that case, like you really can't help. So we try to design up to 10 probes for moderate or rare transcripts.
Okay.
But there are some which are like just 600 or 700 bases long. In that case, we just design like 5 or 6. Yeah. We haven't tried like crowding them to see how they would work. We've always like tried to leave some space in between. Alright, so
I have another question. Do you have any experience with the cell membrane protein USAID? Because you want to raise the cell boundary, right?
yeah, so we've tried a couple of things. We've used lectins. I think one of the images that
Okay.
Bear was showing, I think, had lectins.
That
Yeah.
that you preciously expressing all cell types or just the cell type you're interested in?
No, that's just gonna label all membranes.
Oh.
Yeah,
Oh.
yeah. You can use like lectins, or we've also been trying like cocktails of antibodies to cell membrane markers. That's like one other thing. Yeah. The problem that we've had is they usually don't stain the membrane uniformly.
Mhm.
It's like when you have to like assign cells when you do the data analysis, it's not that helpful because it's not a uniform staining on a membrane. Yeah, so. Yeah, for our analysis we did not like taken cell membrane into account. We've done it without. We just like use the nuclei as a reference and try to like mark cell objects and use it for analysis. Okay, I guess so we can be back here at